A warm and a hearty welcome to one and all present here this evening. I, Doipan Shah from the Department of Computer Science and Engineering, am your host for today's evening. Today is the third day of the 1 week FDP on Advancement of Intelligent System and Computation, organized by the Department of Computer Science and Engineering, JIS College of Engineering, in association with the Computer Society of India, Kolkata chapter. I will request Dr. Partho Sarkar, Principal, JIS College of Engineering, for the welcome address. Over to you, sir.
Good evening, all present in this virtual platform. I welcome all the participants, those who have joined in this virtual platform, and at the same time, I know that it has been live in YouTube kind of thing, where also there are participants also. I personally welcome Professor Subhadip Basu of CSE Department, Jadavpur University, on this day for the program that is the FDP, which is going on in our college on the topic Advancement of Intelligent System and Computation. And today's speaker, he will be delivering the talk on Bioimage Informatics, methods and applications. I hope all the participants, those who are present in this platform, definitely will get enlightened by the knowledge which will be shared by Professor Subhadip, and definitely it will enrich them in their knowledge domain. I wish all the best for this program, and again I will welcome Professor Basu to deliver the talk on the bioimage informatics. Thank you all.
Thank you, sir. Now I will request Dr. Dharampal Singh, HOD, Department of Computer Science and Engineering, JIS College of Engineering, to address today's gathering. Over to you, sir.
Yeah, thank you, Dayapan. So first upon, good evening to the principal, sir. Good evening to the today expert, Professor Dr. Subhadip Basu, sir, Professor of the Department of Computer Science and Engineering, Jadavpur University. I would also like to say the good evening to all the participants who have joined here and also through the YouTube live. And the sir, today topic is the biometric bioimage information method and application. And this is a very good topic. And I hope that all the participants who have joined through the YouTube live, they can get the idea from the sir and try to implement their idea in their research work. Because main motive of this FDP that whatever the participant join through the YouTube live, they can get the idea, try to implement the idea, because personally we believe that knowledge always increases on sharing there. So this is the end from my side, and I request the point to start the session as soon as possible. So Dayapan, over to you.
Thank you, sir. This FDP includes expert lectures on theory, applications, and design methods of intelligent systems and intelligent computing. Virtually all disciplines such as engineering, natural science, computer information science, ICT, economics are all covered. The list of topics span all areas of modern intelligent systems and computing, such as computational intelligence, soft computing, including the neural networks, the fuzzy systems, and whatnot. This FDP covers significant recent developments in these fields, both of a foundational and applicational character. The speaker for today's session is Professor Dr. Subhadip Basu, Professor, Department of Computer Science and Engineering, Jadavpur University. He has received his PhD degree from Jadavpur University in 2006 and did his postdoctoral research from the University of Iowa, USA in 2010 and from the University of Warsaw, Poland in 2012. He needs no introduction. He joined the Computer Science and Engineering Department of Jadavpur University in 2006, where he is currently working as a full professor since 2017. He also holds an honorary position as a research scientist at the University of Iowa, USA since 2016. He has more than 200 research publications in notable international journals and conference proceedings in the areas of pattern recognition, machine learning, bioinformation, biomedical image analysis, and a lot more. He has edited 8 books and has 2 US patent grants on image binarization methods. He has worked in reputed international institutes like Hitachi Central Research Laboratory, Japan, University of Iowa, USA, University of Lorraine, France, University of Warsaw, and Hanover Medical School, Germany. He has supervised 8 PhD students and received several major research grants from UGC, DST, and DBT Government of India. He is the recipient of the Research Award from UGC Government of India in 2016 and the DAAD Fellowship from Germany in 2018. He has also received Boys Cast and Fast Track Fellowship from DST Government of India and has also received a few more awards and fellowships. He is the current chairperson of the computer chapter IEEE Kolkata section, a senior member of IEEE and a lifetime member of IUPRAL. We are really honored to have you, sir, with us this evening. And without further ado, I would request Professor Dr. Subranil. Uh, I will request Dr. Professor Subhadip Basu to start today's session. The topic for today's session is Bioimage Informatics Methods and Applications. Over to you, sir.
Thank you for your kind introduction. I'll try to share my screen and please acknowledge whether you can see the presentation or not.
Yes, sir.
It is visible,
Yes,
sir.
it is visible.
The full screen is visible?
Yes, yes.
Yes, sir.
Yes.
So I'll try to minimize this. Okay, just a second. Okay, so once again, thank you very much and very good evening. And today's discussion topic, I should say, like it's a faculty development program, so it is more like a open discussion. So if you have any queries, wherever you feel, you can just interrupt and you can have questions. So the topic of today's lecture is bioimage informatics. So I'll discuss some of the methods and applications. I hope I am properly audible, and if you have any difficulties, so
Audible, sir.
okay, thank you. So just interrupt. So starting with the very basic definition, so I just took it from Wikipedia that what is bioimage informatics? So bioimage informatics is basically a very new and upcoming subdomain of bioinformatics in a broader sense and computational biology. Okay, so there are many different aspects which are coming up recently that involves the use of different advanced computational techniques to analyze bioimages. The major reason for those bioimage analysis is like these days you have kind of a phenomenal phenomenal growth in the advanced microscopic imaging. Okay, so we have really a lot of Nobel Prizes coming up. They received like the microscopy instead, and interesting visualizations are actually coming up. So every passing day, we can actually see deeper and deeper into the cellular structures. So this is a huge data. So the biologists are actually struggling with this huge loads of data. And they need new advanced computational models to have a better insight from the data. So that is the major reason why this field actually came up, and a lot of computational techniques to analyze bioimages, especially cellular and molecular images in large scale and high throughput. So this last word is actually of high significance, because high volume of 3D, 4D data, that means time varying 3D data, and you need to analyze a lot of them in a quick time to get a meaningful insight. So we need the quick algorithms that can process large amount of data in quick time. So the existing methods, they were struggling to cope up with this sudden surge of data, I should say. And we need advanced methods. So as I said, like automated microscopes are these days able to collect large numbers of images with minimal intervention. And this has led to data explosion, which absolutely requires automatic, semi-automatic. I should say, I would rather avoid the word automatic processing, and I would say that advanced or smart processing. Okay, so the level of automation that we may need into that depends on many aspects. How much accurate you want, what kind of data you want to process, and to what level. Okay, so more or less you need to have really some ability to process large amount of data. So that is by and large the overall domain of bioimage informatics. And today within that bioimaging domain, cellular and molecular images, so I'd rather focus on our neuronal cells. That is like neuroimage analysis, to be more specific. So that is one of my recent areas of research, and we are working extensively in this domain, and I'd like to present some of my work in this direction. So people say understanding the brain is probably the final frontier. Like people are actually starting to, in medical science, so it is advanced these days. So people are trying to devise many different solutions in many different aspects, like take it from the bone research to like lung research, like the heart research. So there are a lot of advancements in different fields, and in advanced countries there is a huge problem these days. Like if you have a severe, like what you have means what kind of problems you develop over the, when a human being grow older and older, what kind of problems are developed. So one definite aspect is like you have loss of bone tissues, like there are a lot of problems with the bone structures, and there are a lot of replacement surgeries that are being done. So you have that lot of fractures, almost like when in your family, like the elder people, they will eventually have some kind of, hip joint fracture, the neck fractures in the femur. And we have a very good remedy, and you have steel plates implanted, and you have problems in the heart, you have bypass surgery. There are a lot of different, like the stents and everything. So everything is available, and you have some kind of remedy. But the problem is with the brain, because there is a huge problem that when you start to lose your cognitive abilities. Okay, so there are newer neurodegenerative disorders which are coming, and you will see that you will see people who are healthy and fit, and apparently they can be cured with respect to the structure of your skeletal structures. Your heart is fine, your lung is properly functioning, but your brain is actually not functioning properly. So this is a big problem. And like people say that we have very little understanding of the brain, and this is probably the last frontier that we need to conquer. Okay, so there are around 86 billion neurons in our brain, and neurons are connected through synapses, and the neurons communicate through electric signals. And you'll see that this particular guy, like the Elon Musk, he has also, along with the rocket science, so he is also very much invested into the project Neuralink, where he is trying to solve these neurodegenerative diseases by this particular gadget, which is called the link. And he tries to implant that in our brain, and will try to connect that to a mobile app, so that you can actually, if someone is having neurodegenerative disorders, and you can actually have a communication with the mobile phone. So you can, if you are interested, and probably many of you are already aware of this project Neuralink. So this is one aspect, like how we can interface with the brain. But that is not just enough. So there are many unsolved mysteries that revolve around the brain, how the memory works, so why we forget, and how we actually develop and manage stress. And there are a lot of people who die of depression. So there are different types of experiments I'll discuss during the course of time. So how stress leads to depression, and all these neurodegenerative diseases, they often originate from those aspects. Okay, so imaging plays a huge role these days in this neuro analysis. So one aspect is like what I'm saying, like interfacing with the brain. So you place some new electrode in the brain, or you have a cap around your brain, and you have some EEG, and you can actually capture some signals. So that is like, you have whatever you have in your brain, you are trying to get those signals. Okay, there are some electric pulses that is happening all around our brain, and you are actually trying to capture that information somehow, either from outside of your skull, or placing something inside your brain, like a link, okay, like a small device. The other aspect is like to analyze and see what is happening inside the brain. Can we image a brain? How to image a brain to the level of a neuron and beyond, even deeper, very deep inside the neuron. Okay, so we have many different types of microscopy available these days. So nanoscale resolution can be achieved with electron microscopy. And also we have, like in this particular lecture, especially we'll discuss more about confocal laser scanning microscopy and multiphoton microscopy, state microscopy, etc. So all these microscopic, very sophisticated microscopic techniques are available these days. This is a part of a very small part of the neuron, that is a dendrite, and I'll come back to this particular small video again at the end of our lecture. So this video is actually prepared by my lab, and this is part of a Nature publication, and you can see that these are small protrusions in the dendritic segment. These are called spines. The purple protrusions, these are called dendritic spines. So these are very small protrusions that actually connect to the axon terminal of the next neuron, and they receive the neurotransmitters through these small spines. Okay, so I'll show you like what our objectives are and how we are doing this. Okay, so if we go back to the basics, you can see that neurons actually send and receive information. There are two basic parts. So one is called like the dendrites, and the other part is called like the axons. Okay, so and there is a main soma, that is the cell body. So you have actually the dendrites that actually capture the neurotransmitter, that capture the information, and the axons actually send the processed information from the soma. So dendrites gather the information and send that information to the cell body or the nucleus, and there they process the information, and the information is then again passed on through the axon to the next neuron. And what happens that along the dendrite, we have small, very small, tiny protrusions, and these protrusions actually connect to these dot-like structures on the axons. These are called the axon terminals, and these are called the dendritic spines. So when you have a connection, then you have actually an axon terminal connected to a dendritic spine and form one single bond or unit, and the information exchange takes place. So you can say that is a signal transmission takes place. And the number of such connections that define a huge role in the in any mammalian brain's cognitive abilities. So in general, there is a general hypothesis that more connectivity you have, you tend to have more, more information processing abilities. And when we grow older, we tend to lose these connections. Even if you have your neuronal cells active, even if the cell is alive, if it has less number of connections with the neighboring neuron through this spine axon terminal connectivity, then we tend to lose our memory or our general cognitive abilities. So this is the general basics about neuron, and this analysis of dendritic spines and the analysis of spine plasticity is of huge interest to the bioimage informatics research community, and it is a very hot topic, and people are working, a lot of key groups are working in this, and there are several interesting hypotheses here, and one of them is like we try to analyze how dendritic spines change shapes to connect to presynaptic terminals, or this in general means these are called axons, and how they get connected. What is the mode of connection? And many neurodegenerative diseases like Parkinson's, Alzheimer, they directly relate to the loss of synaptic junctions. So sometimes I forget things, and my daughters, when we casually discuss things in our family, and she tells me that you are losing the connections. Okay, so when you actually lose this spine axon connections, then you tend to lose things. So that is the general hypothesis. Now, how the connections are made and how they are lost, so that is a major area of research, and I'll discuss that in this course. And there are many different experimental models, and one of them is stress and depression related behavioral models, and they are experimented on the mouse. So as I showed, like you can see that presynaptic terminals like the axons, they send out these neurotransmitters. These are small neuroproteins. They are actually they are received by these spines or postsynaptic dendrites. So this is the dendritic spine. Now, what will be the ideal shape of the spine to receive such a neurotransmitter? That is the main question. So we try to do some kind of structural analysis. See, we are all computer scientists, and end of the day, I am a image processing expert, so to say. So we try to do pattern analysis, pattern recognition, and image processing along these lines. So all these experiments are really complex in terms of the biological sense, and they are usually done on a mouse model. There are many different experimental stuff, but in general, the experiments that we do here are mainly on stress models, stress and depression models. So how you can actually, how stress leads to depression, and when mammalian brain leads to depression, then what will be the general organization of different spine categories or spine types? So that is the general experimental model, and how did they do that? So there are different artificial stressors. So they have a small mouse, and they put that mouse in a stressful condition. For example, if you reverse the daytime condition, so during the daytime, if you put off the light, and if you reverse the sleeping pattern, so the mouse gets stressed. If you put the mouse in front of a rat, so it gets afraid and it gets stressed. So if you do that for a prolonged period, then the mouse finally gets stressed enough so that it gets depressed. So how do you know that a mouse gets depressed? So for example, mouse likes to lick sweet water. So even if it is stressed, if you give it sweet water, then still it will lick it. In another experiment, the mouse likes to swim in a small swimming pool. So if the mouse is stressed, so for stress relieving, like humans, so the mouse swims in the swimming pool. But if it is so much stressed, if you give it specific stressors for weeks, then it will be stressed, overstressed, and then it will become depressed. Then it will not lick the sweet water. Even if you put it in the swimming pool, it will not swim. It will just float. It will not swim. So when the biologists actually, they understand that the mouse is sufficiently stressed, they sacrifice the mouse, extract the brain, and then slice there are certain processes, the brain slices are extracted, and then the imaging is done. And if you apply a specific chemical, certain experimental chemicals, then you can check whether the brain conditions change on the application of the chemical. So this is a biological system, but I'm just trying to give you an overview of the domain on which the thing is working. This is just a general discussion. Now, with this, our work is, ongoing research is on quantitative analysis of dendritic spine plasticity. So we try to analyze that, especially in stressor models, that how to segment those dendritic spines from an overall image of dendrites with spines and how it changes its shape. Okay, so over time, how much shape change takes place after application of a specific drug. So this is the general analysis, and we want to do it in a quantitative approach in a large scale experiment. Okay, so this is there are several good publications. So the theory we developed was published in Pattern Recognition Journal way back in 2007 actually, but we applied this on this particular domain. It's a 2D span, so we call this method as 2D span, a new method for 2D segmentation, classification, and analysis of hippocampal dendritic spine plasticity. So this work was published in the Bioinformatics Oxford Journal in Bioinformatics. So I'll discuss this, and we also have a method for this available for free download, and biologists are actually using this method. So I'll discuss that. I'll show some of the screenshots. And then we also worked on the first one is a 2D model, and the third one is the second one is actually the 3D model for structural plasticity analysis in 3D. So the theory was published in IEEE Transaction Fuzzy System. So it was published in 2016. And then we published 2 papers in Nature, one in 2018 and another one with the German group from Hanover in 2020 last year. So that is in communications biology. So I'll discuss these 2 methods in brief in this particular presentation. So as you can see here, so this image that you see is the image, confocal microscopy image of the dendrite. Okay, you remember that there is a neuronal cell and you have the dendrites, and because of certain imaging parameters, you do not see the complementary axons. So it is based on it is called fluorescence microscopy. So you just apply certain fluorescent proteins, and only the object of interest, in this case our object of interest is the dendrites and spines, so only that region is highlighted. So the white regions are actually, these are actually dendrites, and you can see these small protrusions, these are the dendritic spines. So this is actually a 3D image, and we have maximum intensity projection over the Z space, and you get a 2D image of the 3D volume. Okay, and also you image this over time stamp. So you apply certain chemical in the brain slice and image it immediately, then we call it a time zero image. And then you want to see the effect of that image, the effect of that chemical, and you image the same slice after 10 minutes and then after 40 minutes. And see, like when you administer a drug, then you see what is the effect. So that drug must have some effect in our body, and you want to measure the effect of after 10 minutes, maybe after 40 minutes. So that is the general biological experimental procedure. So for us, we see that this is a grayscale image, and you have actually certain dendritic spines. And what is our objective? So in this particular work, we try to actually segment these particular spines. So we apply a specific approach which is called 2D digital flow, and we apply that in a specific region of interest, and we call it, so this is the boundary region. So I'll discuss like how we are segmenting these spines and analyze several key structural parameters in those spines. So first objective in that segmented region of interest in a dendrite is to segment the spines from the dendritic shaft. You can see that there is a tube-like tubular structure like a dendrite, and you also see that some kind of protrusions, small humps or long or thin regions that are coming out of that tubular structure. So those are the spines. So how to segment the spines from that main dendritic shaft? So for that, we apply a 2D digital flow based algorithm, and the algorithm is such that you have two different sets of kernels. One is called the K90 kernel, that is a 45-degree kernel, and it is an iterative kernel. It is a flow-based approach. And we also have a K45 kernel, that is a 22.5 degree in one side. So you can actually have even more narrower, like K22.5 kernel can also be designed. So I can show you how it works. That suppose you have a dendritic shaft and you have a spine protrusion like this. So the objective is, like, when you start from one side, let P be the starting point, and the kernel is applied here. So P0, P1, P2, P3. And this is iteratively applied for all these points, so that you have this region covered like this. So you have this region covered from left to right kernel. So as you can see that all the kernels are complementary kernels. So this is K11 and this is K12. So this kernel is for flows in from left to right, and this is from right to left. Okay. This is K11, this is K12, this is K45 kernel. So this is from left to right, and for each such point, the same kernel will be repeated. So you can see that if you repeat the kernel from left to right, this region will be filled. And again, if you put from right to left, again this region will be filled, and this is the intersection of the regions covered by both the kernels. Okay, so this is the intersection of K11 and K12, and this is the segmented dendrite, and we call it as the spine. So what we do, we actually segment the dendrite, we mark two endpoints in the dendrite, and the intermediate region gets segmented, and whatever spines you want to mark, you just click on the spine as a single seed and the spine gets segmented. So this is the general approach. As I mentioned, that it is just intersection of R1 and R2, and this is the region highlighted in this light reddish region, peach rather, the peach colored. So this is the some of the examples how we do this with alpha 45 or alpha 90. You can see that if you mark in these two regions, then this particular intermediate shaft gets segmented in peach color, and then you click on the spines on either side and they get segmented. So there can be many different special cases, like when to use which kernel, and you see like improper use of a kernel can actually lead to certain erroneous segmentation results. So that in the tool you can actually have a option like which kernel you want to choose, and by default we suggest that K90 kernel should be chosen. And as you can see that in a specific region you can have multiple seeds, and then you can have all the spines marked in green color on the dendritic shaft. And when you mark the spines, there are actually 4 different types of spines that can be extracted from the dendrite. That is, one is called the stubby spine, another is mushroom spine, then there is a philopodia, and then there is a spine head protrusion. So what is the general biological concept? Again, I'm going to a bit of biological concept that normally you have simple hump-like structures. See, these are small hump-like structures along the dendrite. And when you have some kind of cognitive activity, that means you want to remember something, or some cognitive activity, or you have excessive stress or something, then these small humps, they get elongated. So these small humps are called the stubby spines. And then what happens, these small humps, they get elongated and form some kind of longer structures, and these are called philopodia. And what happens, these philopodia, they look for complementary axon terminals. They try to get connected to the next neuron by looking for a complementary connection. And that can be done only when you have an elongated structure, and the structure literally moves from here and there. If you get a chance to look into the microscope, then you will see that in a brain slice, it is almost in real time. The philopodia is actually moving, changing shape and going from one region to another region. It is like probing different regions for a complementary axon terminal. And when it gets an axon terminal, then it takes a mushroom shape. It takes the shape of a mushroom. That means it tries to see quite naturally that if you touch another object at the tip of a philopodia, then the connection region is less. So the amount of neurotransmitter that can be transferred will be less. So it tries to increase the surface area of contact. and the top takes a head like a dumbbell kind of a shape, and it is called a mushroom kind of a spine. So when there is a mushroom kind of a spine, then invariably there will be a complementary axon terminal, and there will be some kind of information exchange. So another biological hypothesis is like looking for the mushroom spine population. So when we tend to forget something, when we grow older and tend to forget things, that means your mushroom spine population is actually decreasing. And when you try some new drug to stimulate your brain activity, so that is tested on in a mouse model, and they apply the drug, and they will see that whether after application of the drug there is a increase in mushroom spine population. So if we somehow can see that there is an increase in mushroom spine population, then you can biologically validate the hypothesis. Okay, so there are so many unknowns here. So there is also another spine type which is called spine head protrusion. So you have some kind of a mixture of all these three, like some kind of a stubby shape and also some kind of elongated shape. That is called spine head protrusion. So when we structurally analyze, see, analysis of these structures like these mushroom spines and stubby spines. So analysis of these structures are extremely important. So we are one of the among the very first groups who tried to compartmentalize the structures of the spine. So we defined several key points on the structure. So we propose several key points on the structure. So we call this as central base point. That is the point on which the spine is actually connected with the dendrite. We also have a central head point. So when we have a head of the spine, we call it as the locally deepest point. So we have a locally deepest point, and there can actually be multiple locally deepest points. So we can actually have the mean of all the locally deepest point is called the central head point. And there is also a point we define on the spine that is called the farthest point, or FP. So this is used to estimate the length of the spine. Quite naturally, the length of the spine is estimated from the central base point to the farthest point. Okay, so you can see more clearly here. So if you have the dendrite and the spine protrusion is going like this, so this is the central base point, and this is the farthest point, and this is the central head point. So there can be multiple locally deepest points. So you can use fuzzy or simple distance transform. Many of you are aware of like what distance transform is all about. So you can have the distance transform, and there can be multiple locally deepest point using distance transform. So the central head point is actually the mean of these multiple locally deepest points. So you can actually mathematically define that. And what is the length of the spine? The length of the spine is the distance from the central base point to the farthest point, but definitely it is not the shortest path. It is the shortest, it is the minimum cost path through the distance transform map. So you have the distance transform map of the spines. From this is the background, you have the highest distance along the center. So you try to find the optimum cost path from the CBP to FP via the distance map. So there are many important criteria. So this is central point to the farthest point. We actually, using this, we define the boundary of the head and the neck. So this from this side and this from this side. So this defines the boundary of the neck, and this becomes the neck of the spine. So this is really interesting why this behaves much like a transistor. So these spines hold the key to any kind of cognitive activity. So the biology is so unique that these necks, often you will see that neck width is getting altered when you want to, because see, neurotransmitter is coming from this side, from the head to the dendritic shaft for the communication to the soma, that is the central cell body. But when it is coming from here, if you just squeeze this neck, then the neurotransmitter won't be allowed to flow. So if you release the neck, then the neurotransmitter will be allowed. So neck width, this is the neck of the spine, and neck width also plays a huge role in controlling the neurotransmitter flow. It is just like adjusting your weights, or in a transistor, like you alter the voltage, and you how much electrons should be exchanged between anode and cathode, so that can be tweaked. So this is just like your transistor in a mammalian brain. So this is the key structural points in the spine. We will actually utilize this in many of our subsequent discussions. But in general, these are some of the results from our experiments. So you can see that the spines are marked, and there are a lot of spines, and you can mark them very easily. And you can also see there are different colors. So these yellow are the mushroom spines, and these blue are the philopodia, and the reds are the red circles are marked as the stubby spines. Okay, so you can actually, this is the tool that 2D span tool that we actually developed, and this is available for free download by the scientific community. A lot of people are actually using our tool, and that is a very happy situation for us. And you can very quickly mark all the spines, and not just mark the spines, you can immediately classify the spine category and estimate different very important spine attribute, like the length of the spine, the head width, neck width, area of the spine, etc. And we also did multi-user reproducibility, because this is a user interactive software. So you can try with many different users. So the same data labeled by multiple users, and you can see that the reproducibility analysis, what is the result in with respect to area at time 0 and time 10, 2 timestamps we have seen. And in some cases you can see that spine number 34 and here around 101. See, there are large variations. So some people marked it in a different way, another person marked it in a different way. But in general, you can see that the variations are less. There are more or less, you have convergence with respect to multi-user reproducibility, 3-user reproducibility we did. Now, this is the very important aspect, which is called the plasticity analysis. So how much means when you classify a spine, you also need to analyze the plasticity. That means how much is the change in shape? How much stubby spines are changing into mushroom? How much stubby spines are changing into philopodia over a time interval T0 to T10? Okay, so we have used 4 different classes in this particular experiment: stubby, mushroom, philopodia, and spine head protrusions. And you can see that over 10 minutes, T0 to T10, 21% spines more or less stay in the same position as filopodia, as stubby. 17% remain as filopodia. Okay, and you can see that 8% spines turn from mushroom to filopodia. So you are losing the connectivity. And in some cases, you see that almost 5% spines turn from stubby to filopodia, 5%. So this is a 4-class reproducibility analysis, and you can see that spine populations are more or less, there were say 31% spine at in the baseline time 0. After 10 minutes, you see around 35% spine population. So there is no hypothesis can be established that something meaningful is happening, because even filopodia is 32 and it is 33. Mushroom was 27, now it is slightly less, 23. But when we do it for 40 minutes analysis, so T0, T10, T40, so when we extend our analysis up to 40 minutes, we have a very interesting observation. You see that after 40 minutes, you at baseline, you had 48% stubby spine population. So this is a different experiment for 3 times time. And you see that at time 40, you have that stubby spine population gets reduced from 48.7 to 33.3. And the mushroom spine population is changing from 17% to 35%. See, stubby filopodia was 23% to 30%. It has changed. That means 10% spines change from stubby to filopodia. Okay, so this is the highest. That means it is looking for new connections. And then there is a significant percentage that filopodia to mushroom. Okay, that means you have mushroom spine population is actually changing a lot. Here you can also see that there is a significant number of change from stubby to mushroom. So overall, there is a change from the stubby spine. That means you are, after applying a particular chemical, you are actually increasing your mushroom spine population. So this establishes the hypothesis that on a given experimental setup, you can actually increase the mushroom spine population, and you can quantitatively analyze that plasticity. So that is the most important part, that we could actually analyze this in a quantitative aspect, because the biologists had a perception, but you need to establish that in a quantitative sense. So we also did some comparison with the state of the art. There were some ground truth data and some existing analysis. And overall, we can say that digital flow-based approach successfully segments the dendritic spines from the dendrites, and multi-user reproducibility is high. And it can also be extended to 3D analysis. So I'm going to the 3D analysis. I'm not discussing the same biological questions again and again, but in general, the objective is more or less same. Only we try to do this in 3 dimensions. As I was mentioning, that we were taking a 2D projection from the 3D volume, because all the microscopic, confocal microscopic data, they are actually in 3D. But handling 3D data is really complex. It takes more time, very difficult to analyze. So people usually take a 2D maximum intensity projection and do that. But you see that when you do a maximum intensity projection, you lose a lot of details. This is the image in 2D MIP, and you see that this is more clear, na? You have very detailed information of the actual structure, and we try to analyze on that. So the approach that we take here is it is called a shared intensity space. We assume that the intensity in the dendritic shaft is high because the saturation of the dendrite is more when you actually image the data. Okay, so in most cases, you have the dendritic segment having the core of the dendrite is having the highest intensity region, and we call it as a pure dendrite region. And the rest of the intensity band is actually shared space. It can be either spine or it can be dendrite. So we use these as dendrite seed, and then the spine seeds are marked. So this is directly I'm showing you like how we generate the results. So this is one of the results of 3D segmentation. And the basic theory revolves around the concept of fuzzy distance transform, because we consider the image as as I showed that this is a shared intensity space, and in the shared intensity space we have one region as the background and one region as the pure intensity and pure dendrite intensity, and rest we call it as a just a membership function we assign, and a fuzzy subset is designed in that. So we call it as two fuzzy objects as calibrated O spine and dendrite, and we apply a multi-scale opening algorithm in that. So the algorithm, I'm not going into much detail, so it is available in our IEEE transaction on fuzzy systems of 2016, and also this conference paper. But in general, the segmentation objective is to separate the separate the object spine from the object dendrite. And the main beauty of the algorithm is that conjoincy between the spines with the dendrites are actually at different scales. So we can, this multi-scale approach works beautifully well because of this inherent nature of the scale. So nature of like the varying scale. So the scale varies significantly. And with that, we also redefine in 3D the structural element, the structural features like the base of the spine, the central base point, the central head point, and the farthest point. So this is more or less same as the 2D definition. And you can see that the same way you can mark the central base point. This is the farthest point. This is the central head point, and this is the segmented structure. We also did comparison with the ground truth. In some cases, you can see that there are, in case of volume, like in 3D, 3D segmentation is a bit tricky, and there are often some variations with respect to the volume, especially in some of the spines. But in general, if you see the length gives more or less consistent with respect to the manually annotated the ground truth. And also we compared with, there is a 3D segmentation tool, Imaris. This is one commercial tool available, very expensive. But you can see that this tool is also unable to segment the spines properly, and the segmentation, because the structures are so thin, it is not properly, the proper shape could not be analyzed. So this is a comparison with the commercial tool, and some of the images of the segmentation results of different categories of spines. So you can see that these are the stubby ones, these are the mushrooms and philopodia, and these are some of the spine head protrusions. Reproducibility analysis between 3 users, you can again see that in the volume there are certain variability, but length is more or less consistent. And also you can see that plasticity, that is the change of spine shapes over baseline 10 minutes and 40 minutes, you can see the changes. More specifically, you can see that one spine number, it is changing shape, and the change of shape like this is going to spine head protrusions. So this can be very efficiently monitored in 3D. And over the complete experimental scale, you can see that the average change of individual spine morphology, and we can see that there is a contraction in volume, average volume. So initially we had some volumes. After 10 minutes, the volumes are actually increasing, and then there is a slight decrease in volume. So in all the cases, we observe that after apply of the chemical, the best, there is a slight contraction in the average volume, length, and head width. But in general, there is a increase from baseline to 40 minutes. So baseline to 40 minutes, there is an increase. So this 10 minutes timestamp may not be a representative one. We also saw this in case of 2D, that the 10 minutes timestamp was not really a representative one. And maybe there are some amount of random behavior associated with the 10-minute timestamp. We also had data from Hanover Medical School. This is in vivo data. That means this data is from the live mouse. So they have a very interesting experimental setup where you can, instead of sacrificing the mouse, you can actually put a hole on the head, and you can image it in the living condition. Okay, so in a living mouse. So this is a 3D reconstruction of the data, and some sample spines we marked on that. And we showed that even this is called the in vivo data, and live data also, our method could actually segment the spines in a very effective way. And this is the result from this live imaging data. And this is, we published in another Nature paper in 2020. And we could show that one of the very first methods that could actually segment the spines in such a detailed way in vivo data. Accurately, we could segment the spines and mark the spine, quantitatively analyze the structural change of the spines. So that is the most important aspect. And I'm just summarizing this. So overall 3D span algorithm successfully segments and extracts key morphological points on each segmented spine. And the objective is to accurately estimate length, head width, neck length, neck width, all these features. Many of you are aware of digital image processing. You work in digital image processing domain, and this is called digital topology and geometry. We apply different features in that domain. And also for spine classification, nowadays we are working on many different approaches for using deep learning, and for initial pre-processing also we are using several machine learning approaches. So all these things are there, so I'm not going into the main details, but in general you can actually estimate all these structural features, and spine plasticity can be analyzed accurately, and it is reproducible. So multiple users can use and then still get consistent results. and you can download this tool from the website here. And this paper was also published in 2016-17. There's an HR publication, and we got really interesting responses. And like I was very pleasantly surprised when Catherine Willing mailed me, and she is actually from the group of Stefan Hall, who actually got the Nobel Prize in for the state microscopy, the main inventor of state microscopy, and from the Göttingen. And in that paper, that is the seminal work by Stephen Hall. Catherine was the first author, so it was during her PhD. So Catherine was one of the key members of Stephen Hall's group. And Catherine mailed me, and they wanted to use this 3D span tool for their work. I also received mail from Tahare, is a postdoc from the Erwin Nehar group. Erwin Nehar is also very well-known Nobel laureate from same Göttingen group. And also like Daniel also mailed me. Daniel Rubin is a very famous professor from Stanford and director of the biomedical informatics, and they are also testing our tool. We are also, I haven't mentioned in this slide, we are working on a completely new version of this software. And because of there's a huge response that we got for this 3D span tool, and many people are actually using, and we are getting very good responses. So we are completely changing the tool to make it more user interactive and combining different 2D, 3D, and also the 4D, like the time varying data analysis also we are combining in the same tool, and we are calling this as GENIA, that is Jadavpur University Neuroimage Analysis tool. So that is our current project. And I must acknowledge my group, like my international collaborators, Jakub Vlodarczyk and Andre. They are mainly my collaborators in the biological side. Professor Poonam Saha is my main collaborator from US, with whom I actually developed the 3D segmentation method. And Evgeny is also like from the Hanover, he is the group leader, and we work very closely with them related to the data. And Dariush is also, like he was my host for my second postdoc, and my first postdoc was with Professor Saha. And during my second postdoc actually, I got inclined into this bioimage informatics domain, and we started working together. And I must also acknowledge my students, and Pranati is there. Pranati is also, she worked very intensely with me, and we developed many interesting methods on biomedical image analysis, especially related to fuzzy distance transform and also flow analysis. So she also helped me a lot during our bioimage analysis research. And also Shatodal helped me regarding the 2D analysis. Atullah is also there, and currently Nirmal and Shobhik are my ongoing PhD students in this domain, and there's one very good undergrad, and he is developing the GENIA tool. So thank you very much for your patient hearing, and I am sure I'm a bit over time. Like, please let me know if you have any questions or anything.
Thank you so much, sir, for such an enlightening session that we had. and you have really made this evening so special with your. Uh, so we have a few questions in the chat box. These questions have been posted by the participants who are present. So the questions are here in the chat box. Would you like me to read them, or?
These are the messages, right? Let me see. Can we use RCNN for 3D images in the model? Yes, actually, machine learning can be used in this, but there is a big problem because of the availability of the ground truth and labeling of ground truth data. So we are working on certain methods which are very closely related to that, but segmentation of these structures are slightly complicated, and without prior we tried many different approaches. It is not giving the best results. And one very important aspect that we must keep in mind that here one requirement from the biologist is like not to have a completely automatic segmentation. So they also like to have a semi-automatic approach, and they would like to mark the spines one by one to analyze the plasticity. So if you mark a spine at time 0, spine number 1, it must be the same spine number in the next image at time 10 or time 40. So you'll have to manually identify the spine and click one or put some seeds for the spines. So that makes another challenging aspect in automatic segmentation, because how to maintain the numbering. Because when you segment the spines, then it may get a bit randomly numbered, and the spine plasticity analysis becomes a bit challenging. So this 3D span method is semi-automatic, and we use digital topology based segmentation, like multi-scale segmentation algorithm is actually used. For classification, right now in Genia, we are using actually simple decision tree, and in some cases also support vector machine we are using for spine classification. But definitely you can we can use other models. When the dendrite collides with each other, what will happen and which model will be used? This is actually, I'm not sure whether you understood this, means from which angle you actually asked this question. Is it about colliding of dendrites or colliding of spines? So this is actually a very good question, and I briefly showed some examples. So there's a very, there are certain situations when the spines actually touch each other and they become conjoined, or we capture the image like that because of the limitation of the optical resolution. We do not see the difference in our imaging, and they appear to be conjoined. So segmentation of those spines are extremely challenging. Means to segment them as two different spines are also challenging. If you talk about colliding of dendrites, yes, dendrites also, it is a hugely dense network. So there are a lot of dendrites that come one after another. It's a huge chunk of data. We often call it as highways. Okay, so like flyovers, like one dendritic shaft going up and down, and in 3D you can actually visualize that. In the 3D, that the image that I showed you, you have probably seen that there are multiple channels. So they also often touch with each other. different dendrites touch with each other. So for dendrites, since we are not segmenting individual dendrites, so we often ignore that situation. But when the spines touch with each other, then this is a serious condition, serious situation. For 2D analysis, we have certain manual segmentation method. We do that, we handle that manually. And in 3D, there is an option called separator that we use in fuzzy space, and some fuzzy separators are used. That is also manual. So we see, I'm telling one real situation because we are developing a software. This is not always for a research purpose for paper publication. This work that I'm presenting here, they are developed for the biologists, for the use of the biologists. The tool 2D span and 3D span, if you search, you will see that our website is there and you can download the tool and you can use the tool. You can reach me there and you can, people are regularly contacting us that, see, this is not happening. They send us images and show us like this is the problem. So we must also have, along with some sophisticated algorithms, you also have some quick fix solutions so that at the end of the day, they get the estimates. They get the estimates done. So that is one of the reason we, like when designing the overall plan, we, instead of going into a theoretically sophisticated fully automatic approach, we chose to go for a semi-automatic approach so that we have more control. We have more control on the accurate estimation of every single dendrite. We have the control on the length of every single dendrite. But again, at the same time, we can also claim that it is 100 times faster than manually annotating every spine. You won't believe the biologists, for the sake of accuracy, the biologists, they manually draw every single spine. They manually draw and count and estimate the length, volume manually for every single spine. So that is the real situation till date, because the softwares are not easily available in this particular domain for this level of accuracy and that people can actually use. So that is one aspect. So when the spines collide, we sometimes use manual separators. Okay, just a minute. How fuzzy is different from Bayesian network? You can actually look into the paper. It is I can give you the reference of the paper, and you can see, like, I'm not sure exactly from which angle you are asking this particular question, but in general, why we are using fuzzy approach is because we are working in a shared intensity space. In the first 2D approach, we worked on a binary image. So the image was getting binarized. In the 3D approach, we were not using binary image. We were using the image intensity, and the image intensity is within the range of 0 to 255 for 8-bit images. It can also be for 16-bit images. But when you are using an intensity, then you need to estimate the membership of a point, we call it a voxel, to a specific object. So when we are calling it as a membership function, then we actually design it as fuzzy sets. Okay, so that is the general reason why we applied fuzzy. Why not Bayesian? I am not sure from which aspect you are actually asking. So I'll better choose not to directly address this question. Okay, if you have any specific query, maybe you can ask me. I can send you. I can discuss like in which aspect you are actually asking this question. Sir, what is the possible issues related to fuzzy distance? Which algorithm is better to extract features? Related to issues related to fuzzy distance. I'm not again, since it is not interactive, it is very difficult to understand from this single statement. But in general, when we are using fuzzy, whether we are estimating fuzzy distance or fuzzy distance transform, in every aspect, there are inherent challenges for using the concept fuzzy. It is advantageous. Also, there are certain limitations because we have certain preconditions that we are assuming that the shared intensity space behaves in a particular way. And we have higher intensity in the dendritic segment. Core of the dendrite is of higher intensity and the spines are of lesser intensity. So we have this preconception in mind. So sometimes it happens that there are some big stubby spines and the core of the spine itself is of very high intensity, as high as the core of the dendrite. Then this fuzzy distance transform and this fuzzy membership concept fails. So this happens when we have oversaturated images. When the images are highly saturated, then these things happen. And then we can also handle this. Then in that case, we extend the range, and in those cases, we do not take any intensity as pure, like we said, we design that the pure intensity range for dendrites as null. Means there is no pure intensity range. We set it that way. And then we also mark seeds for dendrites. Likewise, we mark seeds for spines. And then you estimate the fuzzy distance. And next question is also like, which algorithm is better to extract features from biomedical images? So there are many features, like in this case, like we use different topological statistical features. You can use any of them based on the application. There is no single model fits all approach. So it all depends on, like, if you work with MRI images, like I'm sure like by biomedical, you are thinking about MR or CT images, then it depends on what application you are actually using. Which algorithm is better for using image processing and machine learning? Can you briefly elaborate fuzzy distance? I doubt like I have enough time for explaining fuzzy distance in this lecture, but you can have actually a simple idea of simple distance transform, and then you can actually have membership of an object. So that is the most important part. So in a simple word, you can say that suppose you have two neighboring points, neighboring pixels. What is the distance? Distance is 1. Because if you have one hop, it is a neighbor. So if it is the 4 neighbor connectivity, you have distance value 1. So the hard distance is 1. But if you make it to fuzzy, then you actually need to multiply it with the average membership of the 2 points to that particular object. Suppose I have a pure object and I define a linear membership function, and if I have a intensity value 255, let us assume that I am giving it as 1, membership value 1. But the 2 points between which I am trying to assess the distance, let us assume they are having membership value 127. The intensity value 127. So just think, like, what is the membership of 127 in 255? That is 0.5, close to 0.5. That is 50% membership. So you actually get a fuzzy distance as 1 into mu of first point is also 127. Let us assume that the next point is also 127. So 0.5 plus 0.5 by 2. So the average is 0.5. So the fuzzy distance become 1 into 0.5, that is 0.5. So you get the fuzzy distance as a product of the absolute distance, or you can also say the product of Euclidean norm and the average membership value of the 2 points between which you are estimating the distance. So this is the overall one-liner, I should say. But if you want to go into details, there are many different concepts that you can actually build on top of that. There are many standard papers. You can just search my name and IEEE transaction on fuzzy systems. There are certain papers. So if you mail me or somewhere, you search in Google Scholar, in Google Scholar citations, if you search, then multi-scale opening. This is, I'm sure you will be having this presentation recorded somewhere, and you will see that I mentioned this in the beginning of my lecture. For detailed analysis, you can actually refer to these papers. Okay, so there are several papers. So I'm talking about this particular paper from 2016. Saha, Basu, and Hoffman. So this is, and here you will, if you find this as difficult, you can actually read the references that are referred at different points, and you will find more papers by Professor Saha, and you will have a better idea about this, about fuzzy distance, fuzzy distance transform, and the morphological operations, etc. So if you have any questions or anything I didn't answer, anything else? Hello?
Hello, sir. Thank you, sir. I think there are no more questions left. So once again, thank you so much, sir, for being with us. And now I would like to request Professor Dr. Pranuti Rakshit, uh, Assistant Professor, Department of Computer Science and Engineering, JS College of Engineering, to kindly hand over a small token of appreciation to Professor Dr. Subhadip Basu. I'll request ma'am to kindly give me the permission to share the screen.
Actually, Subhadip sir, you please stop your screen sharing.
Is the screen visible now?
Yes, visible. Sir, kindly accept. It is just a token of appreciation for your valuable means your intense
Thank
speech.
you. I'm
and
digitally
giving us-
accepting the
yes,
appreciation.
for giving us your valuable time. Thank you.
Thank you very much for your invitation, and I hope some people will get interested in the topic, and at least some information could be, some good message could be shared.
Yes.
Okay.
Yes.
Thank you very much.
Thank you, sir, and thank you, ma'am. Now I'll request again Professor Dr. Pranuti Rakshit, uh, Assistant Professor, Department of Computer Science and Engineering, JS College of Engineering, to kindly give the vote of thanks.
Okay, so being one of the coordinator of this FDP, I am very happy to propose today's vote of thanks, because fortunately our today's eminent speaker is my supervisor, Professor Dr. Subhadip Basu from Computer Science and Engineering Department, Jadavpur University. Thank you so much for your giving us your valuable time and for your excellent and informative speech. Thank you, sir. Then I like to thank our thank you, sir. Then I like to thank our managing director, Mr. Taranjit Singh, for his spontaneous and always support for this type of program organization. Then I like to thank our principal sir, Professor Partho Sarkar, for his support to this type of program. Then I like to thank our registrar madam, Dr. Sila Singh Ghosh, for her support. Then I like to thank all the deans, Dean R&D, Dean Innovative Learning, Dean Student Affairs, for their support. Then I like to thank our HOD sir, Professor Dr. Dharampal Singh, for his spontaneous and enthusiasm to organize this type of program. Then I like to thank our all the faculty members and technical staff who are associated with this, and especially Shonali Bhattacharya madam, Devasri madam, and Shomiran Roy for their support in the question answering session. I like to thank Doipayan Saha for his anchoring, very good and spontaneous anchoring. And I like to thank all the participants without whom this program cannot be possible. So I like to thank you all. I hope I have not forgotten any name. So if I forgot, forgive me. But I like to thank all the faculty members of CSE department, all the technical staff, and all the persons who are associated with it, and all the participants. Thank you all.
Thank you so much, ma'am. And with that, we have come to the end of the third day of this 1 week FDP.
And
yes,
one more, and also thank you, Sudeep, for your support in the YouTube session, all the sessions. Thank you. Okay, Deepan, you.
Thank you, ma'am. So with that, we have come to the end of the third day's session. And again, we have the fourth day session tomorrow. So I would eagerly waiting for all the participants to join, and we'll be continuing these sessions tomorrow again. So with the permission of all the teachers and Basu sir, we are ending today's session. Thank you, everyone. Thank you for being there.